Okay, sorry for the delay. Good afternoon,
problem.
everybody. My presentation is about how to improve our possibilities to assess, predict, and control mold risks. So my name is Maaike le Feber, over 20 years researcher and integrator at TNO in the Netherlands. I have a background in biochemistry, and I'm currently working on exposome research and sensor-based exposure assessment. Indoor mold growth is a known problem for decades, which is expected to even increase due to climate change. Currently, there are no threshold values or norms for mold in indoor environment. Causes of mold growth are, in most cases, a complex interplay between building features and behavior of occupants, often hampering adequate interventions. And besides that, buildings are subject to several transitions that require sometimes conflicting interventions. To deal efficiently with these issues, a multidisciplinary approach is needed. TNO combines expertise in the field of building physics, behavior science, measuring and modeling, health risk assessment, and biotechnology in order to develop effective solutions. So in my presentation, I will touch upon how indoor mold is harmful to humans, why health-based threshold values are lacking, TNO's research for accurate and affordable assessment methods, and why modeling to predict and control mold risks is necessary. So, how indoor mold is harmful to humans? Occupants of buildings are exposed to mold particulates via inhalation. These include both living and dead spores, as well as degenerating mold fragments. Prolonged inhalation exposure to these particulates leads to continuous stimulation of the immune system, resulting in chronic diseases like asthma and allergy. Beta-glucans present in cell walls of all molds are found responsible for this stimulation of the immune system. Vulnerable individuals are in particular at risk, and these includes children, elderly, or people with reduced immune capacity, like obese persons, and this group is growing. To assess the generic health effects, the generic health risk of exposure to mold, no specific identification of the mold is necessary. Besides this generic health effect, more specific effects are known. Some molds excrete mycotoxins that can be very harmful. However, exposure to mycotoxins mainly occurs through ingestion, as these molds often grow on food. Inhalation exposure to mycotoxins is less common. Some molds can cause infections, like Candida auris or Aspergillus fumigatus. This means that the mold grows on or in parts of the human body. Especially invasive infections in lungs or brain may be life-threatening. Treatment of these infections is becoming more difficult as molds appear to become resistant to fungicides used in agriculture. It may be questioned how fungicides used in building materials, like paints, contribute to this process. To assess the health risk of mold infections, identification of the mold to which an individual is exposed is required. So, the health effect of exposure to beta-glucans is a generic mold effect, while infections and mycotoxin exposure are health effects of specific molds only. That brings me to the question why health-based thresholds are lacking. In order to relate exposure to health, personal exposure data are needed as well as health data. Standards for indoor mold measurements are available, but their aim is to identify sources in order to prevent degeneration of the building structure. We currently lack standardized measurement and analysis methods to quantify occupants' indoor mold exposure. As a consequence, the sparse exposure data that are available are hardly comparable nor representative. And if exposure to biological agents has as little priority in your country as it has in the Netherlands, then no change is to be expected on short notice. Besides this, it's currently expensive to perform these quantitative measurements, which is another reason why only a limited number of exposure measurements are available. And seeing the individual sensitivity differences to mold exposure, a very high number of measurements is needed to be able to relate exposure to health effects. Further complicated by the fact that the health symptoms are not very specific, no extensive studies are initiated to create such data. So for these reasons, TNO is searching for affordable and accurate measurement techniques that can be used to create data that can be used to define health-based threshold values, to quantify effects of interventions on buildings on indoor mold levels, to feed modeling activities to predict mold growth and the effect of interventions, and to create data to validate such models. So, of course, we started with an inventory of measurement methods, of which you see a summarized version on your screen. I'm not going into detail, but existing methods are either health relevant or affordable, nothing in between, the yellow line. Besides that, in many cases, not all health relevant mold parts are taken into account. Most methods only focus on living spores. Therefore, we decided to start developing a new method. So we have good experiences with an M&A-zyme approach that can be applied in a lateral flow assay. M&A-zymes consist of two parts, as you can see on the left, that assemble upon binding to the target. Once assembled, the M&A-zymes may activate a coloring agent. The intensity of the consequent coloring is representative for the concentration of the target in the sample. This coloring can be made visible in a corona test-like lateral flow assay that you see on the right of the slide. So, to measure personal airborne mold exposure, we need to detect a generic biomarker for molds. Among other, beta-glucans are such a generic marker that are in addition associated with the health effects. So therefore we are trying to develop this system to recognize beta-glucans. In parallel, we are trying to find ways to sample air and prepare a dispersion in such a way that the M&A-zymes can be added on-site to make our method a point-of-care device. M&A-zymes have an inherent signal amplifying property, making them suitable to detect low concentrations. Herewith, also diagnostic applications come within reach, like exhaled breath analysis or swab analysis for infected patients. Our aim is to develop a device that can be used DIY, so do it yourself, including sampling and evaluation of the result from the lateral flow assay. But this much cheaper M&A-zyme assay can also be applied for research purposes by laboratories in a very efficient and thus cheaper way on a high number of samples. However, this development will take years, and therefore we are also looking for intermediate approaches that we can optimize or apply as is right away. So if you are aware of an affordable, reasonably accurate method, please get into contact with us. So brings me to the last question, why modeling to predict and control mold risk is necessary. Buildings are subject to different transitions, and as a consequence, interventions are put on buildings with sometimes unwanted side effects. For example, to make buildings more energy efficient, the building envelopes are post-insulated with, for example, bio-based insulation material. If the ventilation is not taken care of appropriately, or the insulation material has become wet during the construction phase, the likelihood of mold increases compared to the situation before the intervention, which, to my opinion, is pretty much unwanted. Often, mold is a result of poor building conditions combined with uneducated or poverty-based human behavior. Besides that, also misuse of installations in buildings is a commonly seen issue. Wouldn't it be nice if we had models by which we could predict mold growth depending on the combination of building layout, materials used, environmental parameters, and ventilation? In such models, the effect of different use or maybe misuse scenarios could be predicted before they occurred, like switching off ventilation systems because of too much noise, or opening windows while the system doesn't allow this, or drying laundry in the living room while keeping temperature low to save money. Scenarios that are very realistic in real life. In addition to these use scenarios, also the effect of planned interventions on buildings could be simulated to discover unwanted side effects before the intervention is implemented, with the possibility to adapt the intervention if needed. An affordable and accurate measurement device may support the generation of data to develop and validate such models. So we're pretty much open for collaborations on this topic, either in model development or generation of data. And so I'm very much looking forward to see the next two presentations. Thank you very much for your attention.